Okay, so in this short video, we're going to talk about the last macromolecule, nucleic acids. So in nucleic acids, their monomer is a little bit different than the carbohydrates or proteins because the monomer itself actually has three parts. It has a 5-carbon sugar, a phosphate group, and a nitrogen base. So this nitrogen base is the A, T, C, or G in DNA, or the A, U, C, and G in RNA. So this here, when we look at the 5-carbon sugar, there's two kinds. In DNA, it's called deoxyribose, and in RNA, it's ribose. But when I look at both these sugars, I can see that there's carbon, hydrogen, and oxygen. Now in the phosphate group, I can also see it does have a negative charge. DNA's overall charge is negative. But in this yellow circle here, this phosphate group, that is where phosphorus is used in DNA. And then we have our nitrogen base. And our nitrogen base has nitrogen. So when I look now, and carbon and hydrogen, but when I look now at the elements required for a nucleotide, I have carbon, hydrogen, and oxygen. That when a plant built itself, it had to build its own DNA. So when the plant was building its own macromolecules, it got the carbon, hydrogen, and oxygen from carbon dioxide and water. And then it took up nitrogen for its nitrogen base and phosphorus for its phosphate group from the soil. Those are nutrients that the plant can find in soil. So when we look at our nucleotides, there's actually 4 different kinds of nucleotides, the A, T, C, and G, but they all have C, H, O, N, and P. Okay, so let's go ahead and look at how you can build a polymer from monomers. So when we look at how DNA is built, the T nitrogen base pairs with the A nitrogen base, and the G pairs with C. So we call this complementary, actually. And over time, our DNA strands, these nucleotides, will actually bond together between the sugar and the phosphates. So here, all those little red lines represent the different bonds between sugars and the phosphates. So this right here is your polymer. Now in humans, in one cell's nucleus, our DNA would have 3.2 billion base pairs. A goes with T, C goes with G. We have over 3 billion of those in our long polymer of DNA. So when you look at DNA, one of the reasons it's rad is because it is complementary. If you know one side of the DNA, you can build the other. Now, if you also look at this, you also see that one side is upside down. We would call that anti-parallel. All right, so to summarize, when you look at the monomer and polymer of nucleic acids, the monomer is a nucleotide. It has a C, H, O, and P. It has a phosphate group with a 5-carbon sugar and a nitrogen base. And then the polymer, in this case, this picture is of DNA, but you could also have a single-stranded RNA as the polymer as well. DNA and RNA will both be made of nucleotides repeating over and over and over and over again.